a picture of a phylogenetic tree of genes from humans, mice, and fish. And these are all the same type of genes, olfactory receptor genes. These are the ones that get turned into proteins that are expressed in our nose cells that bind to molecules that are floating around in the air and help us smell things like rose. All other sorts of smells that we can detect. It's kind of hard to see because of the light yellow color, but the fish olfactory receptors are shown in yellow. They show up kind of here on the tree, and there are a few over here. Do you see a group of olfactory receptor genes that are more closely related to fish smell genes than the rest? What position on the clock in this radial phylogenetic tree are the genes that are in mice and humans that are most closely related to fish genes? Yeah, about 7 o'clock. So this whole group of olfactory receptor genes are more closely related in sequence to fish olfactory receptor genes. Did you know that fish could smell? They've got noses. But they don't smell airborne odors. They smell water-soluble odors. So one way that you could interpret this tree is, we've discussed briefly, though, 2 critical points. So I'm kind of starting to review for the exam. One point about phylogenies is, are they fact or are they hypothesis? They're hypothesis. So we use these hypotheses to create additional hypotheses to test. And I happen to have been working as a research technician in the group that did this study. We sequenced thousands of olfactory receptor genes, 2,000 in fact, and we saw this pattern emerge when we looked at how similar in sequence all of these genes were to each other. So one hypothesis might be that these olfactory receptor proteins that we have and that are turned on in our noses, maybe they detect water-soluble smells because they're closely related to olfactory receptors that fish have. And that would be the only type of chemical that fish would need to smell. They don't need to smell volatile chemicals that are in the water because volatile chemicals aren't necessarily dissolved in the water. But that's just a tiny fraction of the total olfactory receptor proteins that we, mammals, have. So what do the rest do? What do you think? Yeah, what's a hypothesis? What do all of those olfactory receptors more likely do? Yeah. So up here the suggestion was these probably detect airborne odors. And you may see, if you kind of zoom in, or if you have better color vision than I do, that blue and red branches on this tree are interspersed. So that suggests that there are lots and lots of species of genes that both mice and humans have that are the same olfactory receptor, which shouldn't be surprising because mice and humans probably need to detect some of the same odors, and we share those sequences because we're closely related species, not sister species. But do you see any places on this tree where there are a bunch of really closely related gene sequences that are specific to one species or the other, mouse or human?
1 and 2 o'clock.
So what color are you looking at?
Blue.
Yeah, so there's this huge patch of the pie here that's just blue. So there are a bunch of human-specific olfactory receptor proteins that mice don't have. So it's like in an ancestor of humans, one gene, one olfactory receptor gene, exploded into a family of olfactory receptor genes by duplication. So where an ancestor back in time, let's say time equals negative, minus, at this point, a gene duplicated and duplicated and duplicated and duplicated and made lots and lots and lots and lots of copies of one olfactory receptor gene that are now multiple genes that we currently have in our genomes and in our noses. What do you think those genes do? What smell do they detect?
Pheromones, possibly.
Pheromones? Could be. It's an so nobody knows. This is underscoring the point that phylogenies are often the basis for creating hypotheses to test in the laboratory. So phylogeny is a hypothesis itself, but it also raises some interesting questions that evolutionary biologists, geneticists, developmental biologists, other fields might want to study. So why is there a whole family, a huge group of olfactory receptors that we have that mice don't?
Are we more involved? No, I guess it would be.
It depends on what those genes do. So is there a smell that humans need to smell that mice don't need to smell that would improve their fitness? That is, might these genes, this group of olfactory receptor genes, might they have evolved because of natural selection? Was there a benefit to having all of those genes to us, and that's why we have those genes?
Could it be to detect human hormones?
So it could be detecting human hormones, right? You wouldn't expect these genes to be present in mice if they're used for detecting human-specific hormones. I'd go along with that. Anyway. So the point here is phylogenies, A, are hypotheses, but most importantly, they get created to help inform us or suggest experiments to do in the laboratory, in the field with these organisms. So mostly, don't tell anybody I told you this, mostly geneticists make phylogenetic trees and then stare at them for a long time and try to find interesting patterns to study. We'll probably just do one of these two phylogenetics topics today, the top one, bootstrapping. This is an approach that geneticists and evolutionary biologists use to ask how strong the shape of the tree really is. It's one way to assess the amount of confidence we have in whether or not the tree that's hypothesized is the most likely tree. Multiple sequence alignments we might not get to today, in which case we would do that after the exam, that is next week. Neither of these two topics will appear on the exam. So we've covered all of the material to this point that we'll talk about and I'll use for exam on Friday. But they might show up on the next exam, which would be the final. Do we have any questions before we move into the new topic, bootstrapping? Questions? Issues?
So we're saying that everything we cover today will not be on the exam?
Correct. But I'd still appreciate it if you stay.
Let's take off.
Well, I am recording it, but then I record every class, and most of you are still here anyway, so I appreciate it. But if, like, that's why I made a point to point out that this will be on the final. This is not free material. So here's the approach for bootstrapping. This comes from the idea of booting a computer, which is a term I don't even know if we use anymore. To pull oneself up from one's bootstraps, that is, how do you turn on a computer if it's not already running? If it hasn't already loaded the instructions it needs to load, then how does a computer turn itself on? So that's the weird concept of why they call turning on a computer bootstrapping. It's like trying to pull oneself up with one's own bootstraps. This is a similar process. It's a technique that phylogeneticists use that uses the data that already exist, that have been collected, to try to figure out, do these data always point to the same shape of tree? And your textbook demonstrates it this way. So we're used to seeing these distance matrices. This is a different type. 6 or 5 different species looking at 10 different traits. And these are the values of those traits. So in this case, each column might represent wings or absence of wings. 1 means you have wings, 0 means you don't have wings. The second column is a different trait. So they're looking at 10 different binary traits they can measure in these 5 different species. And 1 means the individual has it, a 0 means that individual or species doesn't. So we could build a distance matrix based on that table. You could ask across those 10 traits, how many differences are there between species 1 and 2, species 1 and 3, and so forth. We've done that in the past. For the exam, and for phylogenetics in general, what would we do if we found a column, like there was column K, where every individual species, I don't know, what's a trait that every species, regardless of which species we're looking at, would share?
About a head.
What about bacteria?
We do with
Ribosomes.
water. Would
Really
be
metabolize?
like water conservation.
Okay, so they metabolize, so they ingest nutrients. So all 5 of these species require nutrients to live. One of the most ancestral, let's see, if this is a requirement that's shared by all living species because they inherited it from a common ancestor, what kind of a trait does that make this? So it's a shared derived trait, which is a synapomorphy. Right? So this is a synapomorphy of all species. We all require energy to survive. We all contain DNA. That might be this column. You look in this organism, oh, it's got DNA. Great. Or a nucleic acid, at least. If you want to consider viruses to be alive, then we can talk about RNA. Okay. What would we do if we were building a phylogenetic tree and we saw a column that looked like that? Every species has this trait. Right before we make the distance matrix, what do? I cross it out. If it's a synapomorphy, that is all of the species you're looking at share it, it doesn't tell you anything about the relationships between the species. So we ignore it. In this case, the 10 traits that they've looked at, none of those are shared among all 5 of the species. So you could build a distance matrix and build a phylogenetic tree based on that table. Here's how you bootstrap this table. So that distance matrix you would make from the table on the top would give you one phylogenetic tree. And then I would ask you, well, how sure are you that that is the correct phylogenetic tree, that it's the most likely tree? So what you would do, not usually by hand, which we'll do here, but using a computer program, would be to bootstrap. So you take a random sampling of the traits. In this case, you use a computer to try to get as random a distribution of these columns as possible. So what's happened is the computer program, or us in this case, look at that list of 10 traits, A, B, C to J. You pick one at random. In this case, the first one that was picked was F. So they took the data that are in column F and started making a new table with that column of data. Then you randomly pick again from the original 10. So what column I was randomly chosen next. So the data from column I then was added. So we're creating a new table of data, observations from these species, but using the existing data, just different combinations of it. So you'll notice, for example, that C shows up twice.
Three times.
Oh, sorry, twice in a row, and then 3 times. And so forth. So step 1, create a random sampling of all of the original data. Sometimes you use one column more than once. Sometimes, I'm assuming because C was used 3 times, some of these columns of data were not used at all. G and H, for example, don't show up in the resampled table down here. Okay. So now we have a new set of data. It's exactly the same number of data points. So we've still collected 10, we've recollected 10 columns of data. We've resampled it. So same size table, you build a new distance matrix, you look at what tree shape that distance matrix suggests. So you make one distance tree from that table, you bootstrap, resample, you build a second tree. You do this hundreds and thousands of times. That's why we use computers. You do this process over and over and over and over again, and then you ask, of all of those 1,000, 10,000, 100,000 trees that have been drawn, what's the one that most frequently shows up? What's the shape of the tree or the structure of the tree that we most often see? That's probably the most likely representation of the relationships between the species, genes, whatever that we're looking at. So this is not a statistical approach. This is really important. This doesn't give us a p-value. It doesn't say we're 95% or more certain that these are the true trees. It's just one way of getting some idea of how well supported a particular shape of a tree is based on the data that we have. So this is a summary of the process. When you see phylogenetic trees, sometimes you'll see these numbers written at nodes. Those are the bootstrap values. It's just the percentage of times in all, however many trees you drew, how many times you bootstrapped your data. This tells you what percentage of times that particular structure showed up. So in this case, this says that 90% of the time, 90% of the trees that were drawn had A1 and A2 as sister species, and then A3 sharing a common ancestor farther back in time. So that group, that shape of that branch showed up in 90% of the bootstrap trees. That's what those numbers mean. And you'll see these when you start looking at primary literature, probably see them for your term papers. You'll see some phylogenetic trees that have numbers written at the nodes. That's what those represent. How phylogeneticists communicate how likely the tree is to be accurate. So let's put this into practice. I'll do an example, and then as I said before, we're going to do a mock exam today. By the way, the study guide that I posted does not have to be turned in. That's just for you. The key is on YouTube, so you can look at the key after you're done taking this study guide if you want to. So we're going to do the first two together, and then I'm going to ask you to do the third part, question 4 down here at the bottom, as a mock exam to be turned in by the end of class. I'm not scoring it, but I will use it to give you feedback on whether or not you were successful today in class in turning in the annotated PDF, as you will on Friday for the exam. So I'm not going to make us rebuild the top distance matrix. This is one we've done before in class. And that gives a tree that looks like this, with these branch lengths. So that's the original tree. The distance matrix, sorry, my handwriting is getting a bit sloppy. It's a 3. Is that better or worse? So now the job that we have to do is to bootstrap, and I've given you in this case the 5 columns to resample from. 1, 2, 2, 3, and 2. So what's the first thing you do?
Get rid of half of the graph.
Pardon?
Get rid of half of the graph.
Oh, right. So yes, indeed. So we're not going to need to fill in that part of the distance matrix. So now let's rewrite, and that's why I chose a small number of species, a small number of characters. Let's rewrite the trait values. So we're going to need...
Are we only working with the 5 letters to the left, or?
So this is what we're going to do. So I've told you the 5 columns of data we're going to resample. Column 1, then column 2, then column 2, then column 3, and then column 2. So I rewrite the species names just to keep everything straight. and then copy the data from column 1. We're creating a new DNA sequence, or set of DNA sequences apparently. We're resampling. This is the bootstrapping step. I actually did this, I did do this as randomly as possible. I went into Microsoft Excel and asked it to pick me 5 different numbers that were between 1 and 5. So this is a random sampling of the original data. So we copy column 1, and then column 2, which was all T's, and column 2 again, which in the original data set was all T's, and then column 3, CTGA, and then column 2 again. Now, those of you that are analyzing as you go have already probably caught something. Is this a very useful set of data to draw a tree with?
No.
Most of the data, over half of these 5 columns, are column 2. Column 2 is identical in every species. It gives us no data about the relationship. It says that they're all related. It's like DNA. It's like saying, yes, these species all have DNA. These species all have DNA. Are you with me? These species all have DNA. Like, fine, I get it. But this is how bootstrapping works. You make random samplings. So sometimes you will have this happen. So when we write the distance matrix, distance from A to B, they differ by 1 nucleotide, C in A and T in B. So the distance from A to B is 1. A and C differ at the same nucleotide, the 4th position. A and D have, what? 1, 2 differences. B and C only have 1 position where they're different. The 4th column again. B and D have a distance of 2, and C and D have a distance of 2. So is this tree going to look the same or different as the original tree? It's got to look different, yes, absolutely, because the distance matrices are different. So it's going to give us a different shape of a tree, which means that our bootstrap values are not going to be 100%. We're not going to see the same structure of the tree, so we already know that our data are not really robust. They don't tell us the same story about the relationships between the species when they're resampled. So what does this tree look like?
A, B, and C. All have the same distance from D.
So A, B, and C are all the same distance from D. Who are the most closely related species? In this,
A, B, C.
they're all equally distantly related to each other. It's a polytomy. They're all equally distantly related. There are no sister species. There's 3, apparently, sister species. So what are the branch lengths between them? It has to be a total of 1 between A and B, A and C, and B and C. So they're all half. So then the branch leading from the common ancestor to D is 1.5 to total 2 between A and D, B and D, and C and D, which we see in this distance matrix. So at this point, this is just step 1. First bootstrap. So we have to ask ourselves, compared to the original tree, does this node exist? Is there A and B being sister species in this tree? Not technically, because what else is happening at that node? C is also branching there. So these 2 trees at that point, they don't look the same. One's a dichotomy, one's a single branching, one's a polytomy. So that means that that position has a bootstrap value of what percentage of the trees does this polytomy show up in? 50%. So that node has a bootstrap value of 50%. It shows up in half of the 2 trees that we've drawn.
What do you mean? Half the trees is the top?
So these, right, so the top 2. So we've got the original tree, this one, and we've got 1 replicate so far. So we've drawn a total of 2 trees, and only in one of the 2 do we see that polytomy, A, B, and C being equally distantly related to each other. So half of the 2 trees, the one that says up here is question 3, has that particular shape of a tree.
Dr. Ross,
Yes, please.
I'm still not understanding.
So can you clarify what the issue is?
So you got your second tree.
Yep.
How did you get one between A and B when there's 2 differences?
Okay, so what are the differences between sequence A and sequence B in the resampled set of data? So in the original matrix, there are 2 differences between sequences A and B, absolutely.
Okay.
After we bootstrap and take random sets of data from version 1, one of those columns, by chance, wasn't included in the new data set,
Which is what the 1 represents.
which is what the 1
Column
represents.
number.
That's right. So now,
So the next one would be 2 weren't included, and so on and so forth. Is that what those numbers mean?
so column 1, right, so we've got data from column 1, we've got data from column 2 showing up 3 separate times, and we discussed that that was basically useless because column 2 tells us nothing about the relationships between the species. And what was the other one? Column 3. So one of the columns that actually has data that tells us about the relationships between the 4 species. The point of this being, we do the random part because, well, how many different traits, it's a question for all of you, think deeply, how many different traits are there that we could measure to try to figure out how species are related to each other? What's the upper limit? How many traits are there? Did I hear 60? Is it higher or lower than 60? Higher. Don't say it like a question, say it like an answer.
It's higher.
Damn right, it's higher than 60. Right? How many, I mean, it depends, of course. It's contextual. It depends on the situation we're talking about. In the case of DNA sequence, our genome is, what, 3 billion base pairs large or something like that? If every letter in our genome is considered, as it is in this case, where we look at sequence alignments, if every letter in our DNA sequence is considered a trait or a phenotype, that means there's potentially, just looking at the human genome sequence, 3 billion different characters or traits that we could look at. Which of the 4 nucleotides do you have at position 1, position 2, position 3? Of course, no phylogeneticist it's not feasible, of course, it's impossible, that's the point. You can't study every trait, you study the traits that are, well, I don't know, I guess we're lazy. We study the traits that are practical to measure. So we only get a limited amount of information about how species look like each other, act like each other, behave like each other, have the same DNA sequences. So the point of this is to ask, one of the points is, did we by chance happen to observe a set of traits that isn't consistently telling us the same thing about these species? So this first bootstrap tree tells us that we happen to have some data that have no information. We randomly sampled column 2 3 times. Column 2 tells us nothing. So on this tree, the second one, which trait are we relying on to give us the information about these species? Which column? 2 columns. So this tree is based on 2 different traits. That's all. How many traits was the first tree based on? So all 5 of those columns in tree 1 give us some information about how species are related to each other, except for column 2. Indeed, sorry, I didn't see that. Yeah, so column 2 is one I would have scratched out. It's all T's. So the first tree is based on 4 informative traits. The second one, by chance, is based on 2. Sorry, how many? 2. Column 1 and column 3. And that's what this resampling does. It tells us by chance, did we happen to start with a crappy set of data that gives us irreproducible trees, or did we start with enough data to give us consistent hypotheses about the relationships between the species?
Testing the hypothesis.
So this is not testing hypothesis, this is just asking how many times do we arrive at the same tree, the same hypothesis about the relationships. So the reason that we do bootstrapping is because we only look at a very tiny number of traits that distinguish individuals or tell us something about how closely related they are. So this is one way of asking essentially, did we choose the right ones? Did we choose ones that are giving us consistent information about relationships? So tree 3 is not, or the second tree, number 3, is not a very good tree. It doesn't distinguish how closely related species A, B, or C are from each other. So that might be an indication that the data we started with maybe wasn't the best choice of 5 traits to measure. And yes, theoretically, this is a weird way to ask whether or not your data are robust or reproducible or not, but this is what phylogeneticists do. So what I'd like you to do, and we've got about 18 minutes left, is to treat question number 4 like a mock exam. So we've seen how to do the data resampling, how to build, you already know how to do a distance matrix, and to estimate a tree. So I'm passing out a PDF that has, electronically that is, to Google Classroom right now, has question 4 on here. So the idea being, practice what we just did. 4, 5, 4, 1, 3 right here are the columns that I'd like you to resample. So based on those 5 columns of data from the top here, write the new matrix of characters, write the distance matrix in here, draw the tree, add bootstrap values. Branch links too if you want. You can collaborate with each other, ask questions, discuss, certainly. That won't be the case on Friday. But this is, again, just to practice this process. This won't be on the exam this time, but it will be on the final. And I'm certainly available to come around and discuss questions or concerns you have about this. I do acknowledge it's kind of a weird thing to do.
Just the 4th one, right?
Just, yeah, don't do the whole thing, just question 4. So the goal is 15 minutes from now, turn something in. It doesn't have to be the answer. 8:50, back to Google Classroom. Annotated PDF, attached back to Google Classroom by the end of class.
Trying to get up